So now next students we will discuss about the repeated sequences. Okay. Now what are these repeated sequences? As all of you know that there are two regions that are present in the genome. One that is the introns, one that are introns and one that are exons. Okay. And the introns that are the non-coding regions. They are the non-coding regions. Means they are not responsible for encoding the protein. And what are the exons? Exons that are the coding regions and they are responsible for the protein synthesis. So around 90% of the human genome is such that it is almost same. Okay, like earlier we discussed the 99.9%. So 0.1% sequences that are different. 0.1% sequences that are different in the individuals. Now, why these sequences that are different? Means why these 0.1 sequences that are different? Because these are containing the repeated sequences. These are containing the repeated sequences. Means if suppose any sequence is there and any repeated sequence is present and the same repeated sequence it is different in the two organisms. Like suppose this is the sequence having A, G, A, G. Okay. Suppose this is the repeated sequence and in one organism it is present at one locus, at one gene locus, but the same repeated sequence it is present at the different gene locus in the another organism. Okay, so in this manner that these repeated sequences they differ in their position or even in their base sequences, and otherwise the other sequences that are same. Okay, 99.9% sequences that are same and only the 0.1% sequence which is different, that is because of these repeated sequences which are present in the human genome and their position and their base sequence it varies. Now there are two types of the repeated sequences, one that are the mini satellites and the second one that are the micro satellites. Now what are the mini satellites? Mini satellites means that the base pairs it varies 15 to 50 base pairs. Means any repeated sequence means a whole human genome. Suppose this is the human genome and the microsatellite which is of 15 to 50 base pair. Suppose this sequence in the human genome it is of 15 to 50 base pairs, means 15 to 50 base sequences that are present. Okay, now this sequence it is repeated 100 to 1000 times and this sequence it is repeated 100 to 1000 times in this human genome. So this is known as the mini satellite. Okay, means those repeated sequences which are present 100 to 1000 times, and what is the length of these sequences? Around 15 to 50 base pairs. Now next is the micro satellite. What is microsatellite now? It contains less than 10 base pairs. It contains the less than 10 base pair. Means this repeated sequence it contains less than 10 base pair, and this sequence it will repeat 10 to 100 times. It repeats 10 to 100 times. And these repeated sequences that are inherited also. Okay, means from one generation to next generation they transfer. Okay, so this is a part of the satellite DNA. Mini satellite, micro satellite, so these are also known as the satellite DNA. Having means satellite DNA is that DNA which contain the micro or the mini type of the repeated sequences. Okay, if suppose any segment of DNA it contain the mini satellite and the micro satellite, then that segment it is known as the satellite DNA. And these repeated sequences that are also known as the tandem repeats. They are also known as tandem repeats, or you can say VNTRs, variable number of tandem repeats. Okay, so these tandem repeats means repeated sequences that are inherited. So on that basis only the DNA fingerprinting means it occurred. Okay, because like in DNA fingerprinting, if the parents and the child, they have the same VNTRs, means the same kind of the repeated sequence that is present. So it forms the basic principle of the DNA fingerprinting process.